Hi everybody, me again for your final video to watch for day one of our online learning. And the topic I need to explain to you is a topic called RNA editing. I looked and I couldn't find any other videos online that explain RNA editing, so you get to have me again today. First things first, in this diagram here, just a little reminder, this molecule here with the background being, or the back ribbon being golden color, the sugar phosphate backbone, with the bases coming out, this would be our mRNA molecule. And once the cell has completed transcription and has made our little messenger RNA molecule here, the messenger RNA molecule is going to leave the nucleus through these nuclear pores. So this purple ball is supposed to be the nucleus, little circles, nuclear pores, and mRNA is going to leave and head out of the nucleus into the cytoplasm to try to find a ribosome who would be able to read this mRNA and begin to build a protein. But before this mRNA molecule can go to the ribosome and say, ribosome, build me a protein, it needs to be edited. And that's what we have to talk about next. Here we go. All right. So during RNA editing, the mRNA molecule that gets made has to go through the code and identify which regions are the introns, which are these guys right here, the introns, and which regions are the exons. The introns are regions of the mRNA molecule that actually do not encode for proteins. They aren't instructions that the cell needs to build the protein. And since they don't have any point or they don't have any role in helping to build the protein, we want to go ahead and edit these out. We do not want the introns. So in our diagram over here, the blue represents the introns. And as you can see, the cell takes the introns out when they go to do the final mRNA. The final version here only has gold. The gold sections are the exons. The exons are the sections of RNA that do encode for proteins. So we do need these instructions in order to go ahead and create the protein. So we want the exons to stay in the final version of the mRNA molecule, which is why in the end here, all of the exons are there. It's just gold, no more blue. These were introns. They got the boot. They got edited out of the final mRNA version. So kind of a silly analogy that sometimes I think of when I think of RNA editing is say you were going to make some cookies and you're following the steps of the recipe, adding your eggs, your butter, your sugar, and then randomly there's a step in your recipe that said, now stop and do 10 push-ups. You do not need to do 10 push-ups in order to create cookies. You could do them, but you definitely don't need to do 10 push-ups in order to do cookies. That would kind of be like an intron. Just a set of instructions that have nothing to do with making that protein. So they're going to get the boot. They're going to get edited out so that only exons remain. So you don't have any instructions in your recipe for chocolate chip cookies making you stop and give you 10 push-ups. So hopefully that is helpful for you in trying to figure out what this whole RNA editing thing is about. Again, reach out if you have any questions, and I will see you tomorrow on another video. Have a good rest of your day. If I can figure out how to stop it.